Hello, I'm Ana Puiggros from Hospital del Mar, Barcelona, and today I will show you the advances from the European group on chromoanagenesis. This is the outline of the talk. First of all, I will introduce the chromoanagenesis phenomena and a few definitions. Then I will review what is known by OGM regarding chromoanagenesis, and finally, I will explain you the advances from the European group. Chromoanagenesis is a novel class of complex chromosome rearrangements that arise in a single cellular event and lead to the presence of multiple gains, losses, or rearrangements in a few or a single chromosome. It was described after whole genome sequencing techniques, and we can distinguish three types of chromoanagenesis based on the type of abnormalities and also the aberrant mechanisms that generate this phenomenon. The most known is chromotripsis. It's defined as the presence of at least 7 oscillations between 2 or 3 copy number states in few or one chromosome and the associated rearrangements, and is consequence of the non-homologous end joining repair after a massive double strand break on the DNA. The second phenomena is chromoanasynthesis, and is characterized by the presence of duplications and triplications clustered in one or few chromosomes with several rearrangements associated. In this case, the deficiency in the replication fork is the cause of the event. Finally, the third type of phenomena is chromoplexy. In this case, it is characterized by the presence of a series of rearrangements that are chained between chromosomes. It means that the start of one rearrangement is the end of the previous one, and they can present copy numbers in the breakpoints or not. The number of rearrangements is very variable in the literature, and it can range from 3 to over 14 in solid tumors. Well, regarding OGM, the first thing we need to take into account is that most of the studies of chromoanagenesis have been done by whole genome sequencing techniques or genomic microarrays, and that the published criteria could not be directly used for OGM because this technique is different from the previous one regarding resolution or, for example, the type of abnormalities that can be detected. The criteria in the arrays don't take into account the translocations, for example. And finally, it's also important to mention that OGM does not provide information at nucleotide level, so the aberrant mechanism behind each event could not be inferred from the sequence of the DNA. Nonetheless, in the literature, the number of studies describing chromoanagenesis and the potential role of these mechanisms in the prognosis of the diseases is increasing, but most of the studies do not provide specific definitions for OGM data. I will give you some examples of recent studies. The first one is this one from AML, from the MD Anderson group. In this case, they define the chromoanagenesis as the presence of at least 10 breakpoints and at least 2 of these following criteria: clustering of translocations, oscillating copy number profiles, or extensive structural variants, but they don't define specific cutoff in the number of these second criteria to be present. Similarly, the French group published this paper on multiple myeloma and described that chromoanagenesis is very frequent in this entity. And the definition used in this paper is the presence of multiple breakpoints associated with copy numbers and linked translocations in one or more chromosomes, but again, we cannot find any specific cutoff for the number. Another example is this paper published by our group in collaboration with Leuven University, and we distinguish the 3 types of abnormalities and define chromotripsis as the presence of 7 oscillating copy number states with rearrangement, but again, we didn't set any cutoff for the number of rearrangements. For the chromoplexy, the presence of chain translocations, at least 3. And for the chromoanagenesis, we said that it should be duplications and triplications, but we didn't set any cutoff for these numbers. So the idea is that the standard criteria are needed and are mandatory, And then in this setting, we created the European Group on Chromoanagenesis. This group is composed by members of several working groups in Europe. And the first meeting was a personal meeting in 2024 in Montpellier. And in that meeting, we agreed that we need to write down basic criteria as a starting point to start working on the chromoanagenesis field. But that in a second step, we would need to collect real data from OGM cases in order to see if these proposed criteria were okay and works for OGM or not. And finally, the idea is to share the proposed criteria based on the data with additional centers in order to validate this classification and this criteria, and to publish guidelines for the definition of chromoanagenesis for the OGM community. Regarding the initially criteria agreed in that meeting for chromotripsis and chromoanasynthesis, we defined that the minimum number of copy numbers should be 3. That is the same as the oscillation of 7 copy number change. That it was imperative to have associated structural variants in these CMBs. And to distinguish chromotripsis and chromoanasynthesis, the idea is that chromotripsis is mainly composed by losses, while in chromoanasynthesis, we found more gains than losses. Nonetheless, there are several points to be determined, as the minimum size of the copy numbers, if we need to put a minimum number of structural variants associated with these copy numbers, a meaning or a definition for the clusterization of the CNVs, because we think that it's important to have these copy numbers clustered, but we need a definition. And finally, an index to distinguish between chromotripsis and chromoanasynthesis based on the gains and losses detected. For chromoplexy, we decided to set a minimum of 4 chain rearrangements in order to avoid these 3-way translocations that in hematology are known and don't have impact in the prognosis. And we decided that these chain translocations could have gains or losses at the breakpoints or not. And in addition, we discussed that these rearrangements were not exclusively included if they were closed. We also include those rearrangements not closed, because we know that OGM can misdetect these abnormalities, for example, in the centromere or in the telomere, or maybe we can miss some of the derivatives. So it's not an essential criteria to detect chromoplexy. As before, we need to determine still the minimum number of chromosomes involved, or if the copy number variation abnormalities are essential or not, or if we should take into account the presence of tumor suppressor genes or oncogenes at the breakpoints with losses or the fusion genes generated. The next step, that is the one we are working now, is the analysis of real data. The design of this step is the one shown in this slide. We will recruit at least 50 cases to be reviewed. We will review raw data from the red variant analysis pipeline in order to identify in the generated files those abnormalities involved in chromoanagenesis. For this review, we will do parallel reviews by the provider center and a secondary center in order to obtain consensus file identifying these abnormalities. And for difficult cases, we are discussing with the whole group these cases. After that, we have designed an automatic pipeline that will conduct an analysis of these individual files in order to create a global database with the structural variants or the CNBB abnormalities, including only those related on chromoanagenesis for the selected patients. In addition, in this database, we will discard duplicates, and we will check the presence of tumor suppressor genes or oncogenes in the abnormalities. Finally, once the databases are created, this pipeline calculates quantitative parameters of the events identified that will be used to propose the classifier. these parameters include, for example, the total number of abnormalities, the coordinates, the clusterization, the number of chromosomes involved, etc. Now we have been analyzing these cases during the last year, and now we are in the statistical analysis in which we will start the analysis of the median number of these parameters, and we will compare the different events in order to obtain potential criteria to be used for the community. In the last step, we will share the criteria with additional centers from the International Consortium on OGM in order to validate the criteria proposed and to publish recommendations. As we still don't have numbers on these examples, I will show you some examples of the type of cases we are analyzing. These cases are typical cases with chromoanagenesis events. For example, in this case, we have a case with chromotripsis on chromosome 4, 7, and 12 with several losses. And these events also show rearrangements involving chromosome 8 that is a victim chromosome. This case is a typical case of chromoanagenesis in which we can detect several gains clustered in 11q arm and also associated with several rearrangements. And finally, a typical case of chromoplexy in which we can detect in this plot with involved chromosomes the chain translocations between 4 chromosomes involving 6 breakpoints and showing losses at the breakpoints. But not all is as easier as I have shown, and we have several cases with doubts and in which we need to validate the criteria, and that has been left to the second step of the validation to see if we can finally classify these events. Some examples have this type of doubts. In this case, we see that chromosome 15 show a pattern compatible with chromotripsis with oscillating copy number states. By microarrays, probably it would be classified as chromotripsis, but as OGM provides structural information of the case, we can see that we only identify a single translocation in this chromosome. So our doubt is, if we only have a single translocation, is there enough structural variant complexity to be classified as chromoanagenesis or chromotripsis? We will need to check this type of cases. Another type of doubts that we will face is examples like this case, in which we have a chromoanagenesis event on chromosome 18. In this case, we detect more gains than losses, so the feeling is that probably it could be a chromoanagenesis. But as the gains involve the whole chromosome, the doubt here is if we have a chromoanagenesis, or maybe we have a chromotripsis with several losses that is masked by a whole gain of the chromosome, by a trisomy. We should take into account this type of abnormalities also for the criteria. And finally, we also have some difficult cases like this one, in which we detect at least 3 copy numbers on chromosome 3, but these copy numbers are associated with rearrangements that are chained. We can follow a clear chain between the copy numbers involving the loss that then goes to the 10 or the chromosome 13. Here the doubt is it could be a chromotripsis with a translocation involving the chromosome 10 that is only a victim of this event, or are we facing a chromoplexy event with 3 chromosomes and multiple breaks on chromosome 3 that show losses? We will need to validate with other techniques or to compare with other cases in order to define if this type of cases can be classified by the criteria. Finally, just as a summary, it's important to mention that the establishing OGM-based criteria for chromoanagenesis detection is essential to characterize these events and also to assess their clinical impact in hematology, malignancies, and in other settings. That the analysis of real OGM data will enable this identification, especially taking into account objective features and numbers. But as we have been seeing, the task is challenging, and we have more cases with borderline abnormalities or uncertain cases that initially expected. And finally, that additional centers will be enrolled in next steps to validate the proposed criteria that will be published in a future. I would like to thank all the collaborators from Spanish Hematology OGM group that provided many cases for this project. Also, people from our group in Hospital del Mar and members of European group on Chromoanagenesis that have been working hard to obtain the results and that have been presented here. Thank you very much. I will be happy to take any questions. Thank you.